I love to call Ada Resnick and David Higgins to the podium. Thank you guys for being here today. Dr. Adam is a co-director of Data-Driven Discovery and Biomedicine at the Children's Hospital of Philadelphia. He is a principal investigator of the Kids First Data Resource Center. And Adam's area of research is cell signaling mechanisms and oncogenes and tumor progression in brain tumors. Dr. David Higgins, he is the program manager at the Kids First Data Resource Center. And his areas of research is bioinformatics and genetic variants. With that, I will pass the word to you both. Adam and David will talk about the Kids First variant search tool, exploring data sets to jumpstart analysis. Thank you both, and I will pass the word to you guys.
Thanks so much, Marcia.
Yeah, thanks so much, Marcia. And while David pulls up the presentation, I'll only introduce for about 5 minutes some of the framing for the Kids First data resources and where we are, and then turn over to David for the remaining of the presentation. But it's always a thrill to follow up in presenting after Philip, in part because Philip is just an exemplar of that hypothesis that Marcia set forth that is the underpinning for the Kids First program, namely that through the intersection from a developmental perspective across pediatric cancers and structural birth defects, we can drive discovery, greater understanding in ways that might be both preventative and therapeutic in nature, and drive more discovery overall around mechanisms, basic mechanisms of disease. And as Marcia highlighted, the data resources themselves, the data sets, have really exponentially grown to a scale that required the Data Resource Center to continuously innovate around how the platform can best support these large-scale data. But importantly, and this is the part that David will highlight, the challenge for us is to follow up on a second hypothesis of the program, right? So if the first hypothesis is pediatric cancer and structural birth defect can intersect and synergize on behalf of discovery and the advancement of care, a second hypothesis is that in order to support that, we can innovate on the way research is performed through new innovations and technology that really highlight the opportunities for that discovery, not only for the EXO-1 investigators who are submitting samples and generating data, but for the research community at large. Next slide. And so really the framing here is, or the question at hand for us as a DRC, is to continuously, in an iterative fashion, innovate around the way that annotated, clinically annotated or phenotypically annotated biosamples that drive new data generation can end up at the impact phase of research, and transforming that data into harmonized information through reproducible resources that are provisioned by the DRC in ways that can drive the knowledge creation process in the way that Philip highlighted, and ultimately change the course of disease or care for children with these disorders or diseases. And to do that, we continuously have to iterate and deploy alongside these data sets new technologies within the platform in ways that address both the size and complexity of the data. Next slide. And we try to do this in a way that supports discovery across diseases in the way that Philip just highlighted. Next slide, across modalities, across disciplines, and then across data resources, which is a phase we're moving into that Marcia just touched. And it's really thinking about that cross-disciplinary framework that we continuously try to deploy new resources that we hope you as a community engage, provide feedback, and we continuously iterate on that process with the goal of accelerating the discovery process across a platform-empowered resource that brings together communities on behalf of a framework of discovery. And so we're really at that phase where sufficient data and enough resources have been accumulated within the Kids First ERC in ways that moves us into a framework where not only are the EXO-1 investigators who have been provided the resources of sequencing are now leveraging that data for discovery, but where we hope the community at large comes together and drive this forward. And so that can happen both through the portal interface, as David will show you, and then one of the really exciting frameworks that I think we are excited to participate in is how these types of resources within the Kids First, not only at the data layer interconnect with other data sets, but at the platform layer begin to interconnect across other programs across NIH that essentially allow connectivity between data sets that reside across different settings within the NIH, but can be still empowered within a common computational environment. And I think that's really an area of innovation that we hope to drive, and you'll see that as part of David's presentation, that cohort building activities that begin within Kids First can then further downstream intersect within a computational environment that supports the intersection of data sets from various sources, including you yourself as an investigator with access to those resources in ways that's newly empowered through some initiatives that you'll hear about a little bit later in the program. These resources are part of a complex environment that David just highlighted it and Marcia highlighted. And so this complex environment is still evolving, still maturing, but it's a rich and fertile area of innovation that we hope any of you who are participating in any one of these efforts or others continuously interacts with us within the Kids First ERC to continuously inform how we might do better and continuously improve on these resources on behalf of your efforts as investigators and community stakeholders within the pediatric landscape. And I'll turn it over here to David, who will go through a use case framework that really starts at trying to leverage, the more than, I think 300 million variants that are currently available for exploration across more than, I think, 60 billion occurrences across these data sets to drive a process of discovery as a model. Back to you, David.
Thanks so much, Adam. Yeah, so thanks for that great introduction. So today I'm going to talk a little bit about the variant search tool. So this is a feature that the Kids First DRC launched on the portal last year that we think is really a great opportunity for folks to be able to compare and search the genetic data across Kids First data sets and able to find these cross-disease comparisons and make these discoveries that we're looking to drive forward. I do want to squeeze in my acknowledgement slide right here at the beginning. So the Kids First DRC, Adam and I are standing here presenting today, but we are a huge group of individuals. And the variant search tool was built by our partners at St. Justine under the team of Dr. Vincent Ferretti. So just wanted to acknowledge them for all their great work putting together this really exciting tool that I'm going to show you today. So what is the variant search tool? So it allows users to be able to search the Kids First genetic data. And so we already have the Explore data tool, which allows users to be able to explore based on phenotypic and clinical data. And we have the file repository that allows users to be able to explore files based on different file types. And so variant search allows you to be able to search the genetic data. And so what you can then do is then you can use this to identify new samples and build cross-disease cohorts. Which goes back to our title, which we hope will allow users to be able to jumpstart these discoveries in pediatric cancer and structural birth defects. So when you go to the portal, you'll have this is what our tool looks like. And I'm going to show some of the features here, and then we're going to go into some use cases where we walk through how could we use variant search when we dive in here in a minute. So when you first log into the portal, we have a main menu up at the top. And you can navigate to the search tool by clicking that variant button right between Explore data and the file repo. Once you click that button, it'll take you to this page. And so over on the left-hand side, there's a column that you can use to apply filters to the database. So you can imagine, as Adam said, we have millions and millions of variants here. And so you can filter down to find specific subset that might be relevant to your research. And so you can apply those filters. Once you do, those filters will appear in this query up at the top of the tool. So here we're looking for variants that are in the gene MYC. and we're screening for where they've been VEP annotated to have a high or moderate likelihood of altering the protein's function. And then below, the results will be shown. And so here is each of the individual variants. So at the particular locus on the chromosome, links out to dbSNP if available, and spells out protein consequences in addition to showing which studies and how many participants that variant is found in. And so to show you some of the features up close, I wanted to pick some examples. And so I actually wanted to build on some recent discoveries using Kids First data to be able to explore and dive a little bit deeper and begin to make these comparisons. And so first I'll introduce this story about cleft palates, so orofacial clefts. So cleft lip and cleft palate are among the most common birth defects worldwide. And Philip did a great job. He showed some GWAS studies, which can be used to identify variations in individuals associated with a particular phenotype. And so, of course, these GWAS studies can also be done on individuals with orofacial clefts as well. And so a study in 2020 from Dr. Mary Marizita's group did a GWAS using Gabriella Miller Kids First sequencing data. And their group identified a region on chromosome 21 in a population of participants and patients from Colombia that were associated with orofacial cleft. And so we could then imagine this question, well, is this variant present in participants in other Kids First studies as well? So I'm going to exit out of my slideshow here. So we can actually search for this variant in the variant search tool. So I'm going to copy that RSID, and I'm going to jump over to the portal. And so here I am in the portal, and I'm over in the variant tab in the variant search. And so we can search, this is a variant, so we're going to use the variant set of filters here. And then I'm going to paste that RSID, and there it pops up. And there appears in the tool. And so it tells us information. Yep, this is on chromosome 21. It can link out to the dbSNP page to learn more about the particular allele. This one is in an intron. It's not affecting the protein. And it tells us some information about the number of studies as well. I can click and learn more about this particular variant. If I show different frequencies, and this variant is actually quite common. It shows them in many Kids First studies. And so it indicates these are our different Kids First studies, and this is the frequency of how many participants in that particular study have this variant. So if I go back to my page here, and so we said this is very, very common. So 5,000 different participants have this variant in their genome, as we identified through the Kids First DRC harmonized data analysis. So I can actually click this 5,000 participants, and it will take me over to the Explore data tool. So now I have a query here where I have, this is the 5,324 participants, and then I can continue to kind of add additional filters to explore this set of participants further. So you can see, for example, the most frequent diagnoses here is cleft lip, cleft or palate, but they also have other diagnoses as well across cancers and other birth defects. So if I look a little bit deeper, I could, for example, narrow this by study. So I could say I'm interested in the kids in the heart defect study. And then I can narrow that further. And so now my frequent diagnoses include various heart defects phenotypes. But I can actually add back, but I'm interested in maybe the kids who have that orofacial cleft. So I can go into the Observe Phenotype, our HPO browser, and type in orofacial cleft, use this term right here, and add it to our query and apply. And so we get to this very, very specific query here. So this is participants with this variant in the heart defect study who also have the orofacial cleft phenotype, and there's 2 participants. And we can learn additional information about them below. And from here, then I could take these 2 participants, explore what files are available for them. And from here, if I'm interested in exploring, for example, the variants, I could select the GBCF files, and then I could move these files over to Cavatica for analysis. And so I'm going to come back to these in a little bit at the end of my talk, but showing how you can use the variant search tool to identify participants, find the participants with that variant, and then move them to Gabriella for analysis. So to jump back to my slides, so that's a single variant. So we looked up one particular variant in the variant search. But we can look more broadly than that as well. And so we can look at, for example, some findings from the Neuroblastoma Study. So neuroblasts are a type of cell found in unborn children, which will develop into neurons. In some children, neuroblasts continue to grow and form tumors, and that cancer type is called neuroblastoma. So neuroblastomas are frequently arise in adrenal glands, but can be found in other tissues as well. And so a study in 2019 from Dr. Sharon Diskin's group found that a small deletion on chromosome 16 was found to be more frequently observed in patients with neuroblastoma. And so using kids' first sequencing data, they helped support that finding by looking at coverage in our trio-based studies here. So here's mom and dad, and then it's actually a de novo deletion in the patient where you can see coverage across that position in the genome is lower. And so this region of chromosome 16 encodes many genes and potentially many variants. So could we do a larger search? Could we search across this entire region of a chromosome and see what variants in that region are present in other kids' first studies? And so yes, we can. So I'm going to go back to our variant search here. And I can even save that query, and I'm just going to do a new query right here. And so we can go back to that a little bit later if I want to pick that up again. So I can search based on by variant. And so we can search based on chromosome. And so this is chromosome 16. So we can select that. And so now we have filtered our variants just to those on chromosome 16. And then I can filter based on our chromosome coordinates as well. So if I put in my genome coordinates, copy and paste right there, and click apply. So now you can see we're looking at chromosome 16, and we're only looking, within that framework along the length of the chromosome. And so what this pulls up, you can see, wow, there's quite a lot of variants. So there's over 700,000 variants. So this could be, a lot to sort through. So we can actually filter this further. So we can say, well, maybe let's just look at the ones that have a high VEP score. So are most likely to be affecting protein function. So we can add that filter, and that gets us down to 785 variants. And so from here, we can then scroll through. So because we're looking at multiple variants at once, these are going to be present in multiple genes that are present in that region along chromosome 16 as identified by that study. So these are rarer variants. And so these are only showing up in a very small percentage or a very small fraction of participants. But you can learn which studies these particular variants are arising in. So if we go over to frequency, this gives us information that these are the 3 different studies with the number of participants that have that variant. And so then an investigator could apply for these studies to receive access and then be able to access that data in the Explore data tool and also push it to the file repo and to Informatics. Lastly, I'll show you, so it can be used to look up existing findings, but it can also be used to make discoveries as well. So we've loaded a lot of existing annotation information into the database as well. So here I'm showing 2 types of birth defects. So on the left, that's an atrial septal defect. And so this is a hole or an opening in the atria that allows blood to flow from the left side to the right side. And on the right here is a congenital diaphragmatic hernia. This is a hole in the diaphragm that allows organs such as the liver to develop out of place. And so both of these are birth defects. And so we can ask, is there an overlap in genes underlying these birth defects? And are there any variants in these genes present in kids' first participants? And so we can answer this first question, are there overlap in genes underlying these birth defects? Because we've loaded annotation information into the tool. So under our pathogenicity tab, if I do a new query, or excuse me, under our gene tab, I can search under HPO. And so this will pull up, so this has our human phenotype ontologies. This is genes that are known to be associated with these particular phenotypes. And so if I search for atrial septal defect, I can apply. So this tells us all the variants in all of the genes in kids first that are associated with this particular phenotype. And I can add a second filter where I'm going to look for our diaphragmatic hernia, and we're going to apply that. And so now this is telling us two separate sets of variants. And you can imagine I want to do like a Venn diagram overlap there. So I want to compare these two and combine these queries together. And so, yes, there is overlap. And so, for example, this gene SM3 is implicated in both septal defects and diaphragmatic hernias. And it gives us information about, what studies and how many participants in these kids first studies have these variants as well. So it allows you to be able to look up and continue that discovery. So that's variant search. And so it allows you to be able to search and identify, variants across kids first data sets. And then once you make that discovery, as I showed, you can push those files from the file repo over to Cavatica, which looks like it logged me out while I was inactive watching the earlier presentation. But if I log back in really quickly.
Maybe while you're logging in, David, I'll just make a comment. The framework within which David is operating right now is a framework that's accessible to anyone, right? So anybody can log into the Kids First portal into what we term the registered tier framework, right? That once you go through a click-through agreement, you can have access to the clinical phenotypic data and then to this information in the variant browser, as long as it meets certain criteria. So you'll see, for example, in the view that David is highlighting now, the studies numbers are linkable, and you can click on them and explore that data. But if you have below a certain number of participants, you will not be able to click on those and go directly, right? So here you have 2 participants. And so without having dbGaP access, you won't be able to actually view those. And so we're really actually, really straddling that informational space where we are trying to allow investigators to explore across a large number of studies in order to assess what studies they might be interested in to take a deeper dive into in the format that David will just show you, right? So once an investigator has access to such data through dbGaP, once they have done the cohort creation exploration, they can operate across that data and other data through Cavatica, which is where David is going to head to next.
Yeah. So these are just to show, so these are my demo project here for the webinar. So these are the VCFs, and so we actually have the indexes that were provided as well. So you can move files from the portal to Cavatica, and then they appear here. And so from here, Cavatica has a couple different features. So if you're doing a large scale analysis, you can build your own app and workflow. But today I'm going to show off the interactive analysis, which lets you be able to access these files here in the cloud. And so, for example, I'll show that it has for example, a command line interface where you can interact and see, yep, there's my VCF files. And so you can review them at the command line and run your any Unix-based tools. But you can also run notebooks as well. And so here, for example, this is actually a demonstration from a talk by the NIH data scholar Andelette Mays, where we're loading data. So you can load R packages, and then by loading your data set, we can then run our command and then begin to do plotting and analysis. This is demonstration data. I didn't want to show the controlled access VCF data today, but it gives you a sense of, you can go very quickly from variant search to the file repo, to the files, and then you can run your analysis on the VCF and begin your graphical output very, very rapidly, all in the cloud. So I see we are approaching the end here, so maybe I'll give one last shout out here at the very end. So thanks so much for giving us your attention today. If you want to learn more about Kids First platforms and data sets, we have a monthly office hours series. And so these are usually on the second Tuesday of each month, but that was yesterday. And so we wanted to move it so that way we could give you all an opportunity to join. And so we've rescheduled the May session to next Tuesday, Tuesday, May 17th at 3 o'clock Eastern. And so if you're interested in joining, there's a link there, as well as a link in the chat. And we're happy to follow up and give more information about how to use variant search, how to use Kids First data sets. And we're happy to continue the conversation after the webinar ends today.